that right?
Yes, sir.
Okay. I'd like to thank Cass for including us this time. I think I can speak for our whole team when I say that we found this whole exercise extremely intellectually stimulating and has really helped us expand our understanding of a project that we started way back in 2015, I think, something like that. And it's really nice to be able to show our results from our experimental side today. So, as my title suggests, I'm interested in presenting a probabilistic view of structural biology and the fact that there's a significant challenge in determining, describing, and predicting the structure of a flexible protein. So we happen to be working with one of those that I'll tell you about, and it really expanded our definition, at least, of what structural biology is. So if we think about probabilistic models of biomolecular structure, the goal of, in general, experimental structural biology is to make useful models of biomolecular atomic structure based on experimental observations. The goal of CASP is to aid in the development of a generalized predictive model that's based on sequence. So this is an exercise in doing both, right? Conventional structural biology can't usually be used to build a model of a flexible protein. I'm going to talk today about a protein that Guy likes to call domain linker domain. I think it's a nice description of our system. And what we're trying to describe between my group, our group, and Susan's group is both the interdomain orientation, that's what we're addressing with NMR, and the distance distribution of a domain linker domain protein. And in both cases, we want to determine this structure as a continuous probability field, where the probability field is on the scale of the whole protein or the domains of the protein. Therefore, the structure of a DLD protein is best described with a continuous probability distribution because, in fact, the two domains are moving in this continuous probability distribution field. We use NMR residual dipolar couplings to provide information about this kind of continuous distribution, and we call it a continuous distribution of interdomain orientation, or CDIO. To tell you a little bit about the system we're working with, it comes from staphylococcal protein A, which is a cell surface protein attached to the peptidoglycan cell wall by this region X linker. It's not really a linker. It's a long extended, poorly conserved region of the protein. But the business end of the protein are these 5 protein binding domains. They bind to lots of different proteins, actually, in the host, not just IgG. And they come labeled as E, D, A, B, and C. The B domain has been worked with as a biophysical and biotech model for quite a while. And there's been a minor variation of B where 2 residues are substituted with alanine, and that has been called the Z domain. That's a biotech tool for purifying antibodies. And it happens that today we're going to be using the Z domain in the first of the 2 DLD protein constructs. Each of these domains is quite small. It's only 58 residues, and the structure of each of them is nearly identical as far as we have structures of, which is for almost all. Well, I think we have one kind of structure or another for all 5 domains, and they all fit this very well, this 3-helix bundle structure. What I've highlighted here are something very important for our discussion today, which is the ends of the 2 protein, which form the linker between each of the domains. And in many ways, the linker is the star of our show. Here's the sequence alignment, and it shows why there's so much structural and functional homology between the domains. It's because their sequence is nearly identical. So you can see that when we line up, all the residues that are grayed out in this picture are completely conserved, and there's slight variation at the other residues. What's most important to point out is the conservation of the linker sequence, which is nearly identical in all 4 cases, although there's this AQQ insertion in the ED linker. But other than that, this is highly conserved as well. Now, you might ask, is this conserved if you were to blast the sequence against the database? And the answer is, it is. All of the 190 or so Staph aureus protein A submissions to the database, to the BLAST database, are 100% identical with this sequence, which shows you how well conserved the N-terminal domain of this protein is. So multiple sequence alignments really would just give you one alignment, at least for the top hits against this protein. It really is fairly unique, and it's only Staph A that has this protein. All the other species of Staphylococcus do not have this protein. What's interesting is that region X is used by the people who categorize and cluster staph strains, various isolates of staph from the real world, from clinical isolates, and they cluster the various isolates based on sequence variation in region X. So what's really striking is region X is highly variable, so variable that it's useful in making clonal clusters. But this region, what we call SPA-N, is highly conserved. The final thing I want to point out about this protein is what really led us to think about it as a domain linker protein. And that is that when we do NMR relaxation experiments on this sequence, what we find is that the order parameters of the core domain, the 3-helix bundle itself, are quite high. This is as high as typically you find in well-ordered and not particularly dynamic proteins, around 0.9 or above for most residues. But you can see that the order parameters for the linkers are really quite low. And that was our first clue that this was a situation where the flexibility between the two domains provides them with both thermodynamic and structural independence. So we set out to try to figure out what the structure of this unstructured region plus the two domains might be. And of course, conventional structural biology is not particularly useful for that. But fortunately, Jem Prestegard's lab has developed a nice system for orienting one of the two domains by putting a lanthanide binding tag in the domain. And fortunately, his group had done that with the Z domain of protein A. So we took their construct and linked it via the conserved wild-type linker to a C domain, the second domain. And that's how we came upon ZLBTC as our protein. The advantage of the lanthanide linker is that it allows partial alignment of the first domain without directly influencing the relative orientation of the second domain. So that gives us the ability to identify what the orientational arrangement pose of the second domain is relative to the first, using the alignments that the first domain can achieve. Here's what our raw data looks like, or semi-raw. This is a so-called IPAP spectrum that shows splitting in the nitrogen dimension that is both the sum of the J coupling, which in the reference domain, which has a diamagnetic lanthanide that doesn't align the protein. So this is an unaligned sample. The J coupling for this particular NH group is 93 hertz, but when aligned, it drops to 86 hertz, right? So that means that there are 7 hertz of what we call residual dipolar coupling. The remaining, that difference between what we see between aligned and reference is the remaining dipolar coupling that is the result of the partial alignment of the first domain. But when we're looking in the second domain, the RDCs that we observe are influenced by the range of motion of the second domain with respect to the first. And the math for doing this is fairly straightforward. It can be found described in detail in Bruce Donald's text called Algorithms for Structural Biology, and I strongly recommend that you look that up if you're interested in the potential use of RDCs as a structural constraint. This describes the global tumbling of the oriented domain and then the internal motions that give us the arrangement of the second domain. And with a fairly straightforward set of linear algebra relationships, we can figure out what the orientation of the second domain is with respect to the first. When we do that and then back calculate the RDCs from the deduced inner domain orientation, you can see that we do a pretty good job of reproducing the experimentally observed RDCs. So these are the RDCs that we see in the oriented domain, the domain with the lanthanide in it. And you can see that they range over a fairly wide range from minus 20 to plus 20 hertz. We're not getting very many, and the reason for that is that the lanthanide in the tag actually relaxes a large fraction of the observed resonances in that domain, and so we can only observe a few, but we get enough so that we know what the orientational preference of the Z domain is. And then when we observe the same kind of thing in the C domain, what we're really observing is how this is oriented relative to the Z domain. And you can see that we have a lot more resonances because we're farther away from the paramagnetic ion, but we also see much smaller RDCs. And the reason we see much smaller RDCs is because of that internal motion that reduces the amount of ordering that produces the RDCs in the first place.
Right. So unfortunately, we're in a very tight schedule. So
okay. You can see that our uncertainties are pretty low, 0.2 hertz. Okay, so how do we represent this interdomain orientation? So here's an example of one unit of one sample of a continuous distribution, and we have to represent it by representing the orientation of the z-axis of the blue domain, the non-oriented domain, relative to the red axis of the oriented domain. And that z prime axis is then shown as a position of each of these disks on the surface of a sphere. And then the x prime axis of the blue domain is shown as the color scheme that you see in each disk, where the color corresponds, the redder color corresponds to higher probability. So we
really need to move on. So if you can really quickly wrap it up, unfortunately. So.
Okay. So let me jump ahead. I'm having trouble moving my slide. So let me just say that having shown those disk on sphere representations, I want to show three what we'll be sending out to each of the predictors, which is a disk on sphere representation of their predictions and how they compare with ours. This is the open complex group, which actually does the best job of capturing the fact that in the prediction T1200, which has the wild type linker, they get a fairly anisotropic prediction. It's not consistent with what we observe, but it's not isotropic, whereas many of the predictions were isotropic for both the wild type linker and for the Glide 6 linker. And for the T13 target, the Glide 6 linker is isotropic. So this is one of the better performing groups. I'll just finish by saying that thanking the people that were involved with this, Alan McBride, Edward Chung, and Elaine Mapuli, were the people who did most of the work on this project. And Yang Chi started it back in 2015. Susan is collaborating with us, and she will talk next. And Ron Venters has helped us with the NMR data, and these are the funding. Thank you.
Thank you very much.